Welcome to AP Biology. I want to talk about two different types of receptors today. We'll talk about ligand-gated ion channel receptors, and then we'll also talk about intracellular receptors. And I want to compare that first one to the receptor tyrosine kinases that we talked about last time. So with this receptor protein, it's both a receptor protein and an enzyme, right? It's an enzyme that phosphorylates the next molecule. With the one that we're going to talk about in this video, the ligand-gated ion channel receptor, this works both as a receptor protein for a ligand and also as an ion channel, which is kind of cool. And so the way it works is you might remember that your body is electrical, right? So like an EEG, electroencephalograph, right? That is a tracing of the electrical pathways. It's a tracing of like the electricity in your brain, whereas an EKG, electrocardiogram, this is a tracing of what the heart electricity looks like. So it would be sort of like beep, 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 right? You might see that on TV when you're looking at someone in a hospital. And so this is the top, the atria contracting, and then the ventricles contracting, and then the ventricles relaxing. Anyway, the whole point is that you are electrical. Kind of just like your computer, except the moving charges in your computer are electrons, and that's not true for you. The moving charges in you are also moving charges. It's just that they're not electrons, they're ions. So ions like sodium or calcium or potassium or the chloride ion. So these are the charges in you. That's the electricity of your muscles. It's the electricity of your nerve cells. And so the way that it works is that you would have a signaling molecule, a ligand, like maybe serotonin, norepinephrine, I don't know, acetylcholine. When it attaches to the receptor right here, it causes the receptor to open up. And so here you have a channel protein, right? It's a channel protein that allows materials to go from where there's a lot of them, from a high concentration, down the concentration gradient to a low concentration. They're going high to low. This is facilitated diffusion. It's passive transport, right? Facilitated one T, facilitated diffusion. And so that's really important for nerve impulses and for getting a muscle to contract. It's the impulse that has to go along a muscle to say, hey, muscle, time to contract. And we actually have sometimes these ions important in the cell too, like when calcium is released from the endoplasmic reticulum. And so when the ligand is off, the gate is closed, and when the ligand is on, the gate is open. So if you take anatomy, you'll find that these are really, really important for the nervous system and the muscular system. So that's really all I have to say about that one. The last receptor that I want to talk about are intracellular receptors. So that means within the cell. So here's that receptor within the cell. Here's the plasma membrane, right? So this receptor is unlike all of the other ones, right? The other ones were right on the surface of the cell, right? We saw them right on the surface of the cell. This receptor is found inside the cell or sometimes even in the nucleus. And so that means that this ligand has to be able to go through the lipid bilayer. So remember, the lipid bilayer is made out of phospholipids. They have little smiley faces that say, I love water. And they sort of all just like hang out together. Oh my God, I love water too. Oh my God, me too. We're all like so much alike. All the heads are saying, I love water. And all the tails are saying, not so much. So the tails are hydrophobic, right? So the heads with their little smiley faces, I'll have to put the smiley faces on after. Okay, so here's the lipid bilayer. Sometimes you'll see it drawn like this. This is probably a simpler way to draw it, maybe kind of like that. So this is what the lipid bilayer is going to look like, either of these pictures. And so let's get another color. Okay, so the little smiley faces are here saying, I love water. So the heads here are the hydrophilic parts. Oh my God, I love water. Oh my God, me too. Oh my God, I love water too. Okay, but the lipid part is the hydrophobic part, and that's really the bigger part. You can imagine that a molecule might be able to get in between these heads, right? They're smaller. But the lipid part is the big part of it, and that's the hydrophobic part. So what can mix in with hydrophobic stuff? Well, other hydrophobic stuff. So let's say you have a signaling molecule that looks like this. It's just chains, it's rings of carbon with some hydrogen sticking off. Let's say it kind of looks like that. That's a lipid. It's hydrophobic. That'll be able to go right through the lipid bilayer, whether you want to draw it like the top picture or the bottom picture. So the molecules, the ligands that go to a receptor that's inside are ligands that have to be able to go through the lipid bilayer. So they have to be hydrophobic, just like the membrane. Any hydrophilic, whoops, any hydrophilic ligands would have to go to a surface protein. The receptor would have to be on the surface. But if it's a hydrophobic ligand, the receptor can be inside the cell, even inside the nucleus. And so here the two are together. It's the hormone receptor complex or the ligand receptor complex. If it's testosterone, it's going to go right into the nucleus and it acts as a transcription factor. It goes to a particular gene, recognizes it, and tells the DNA to make RNA. So we haven't covered transcription yet, but we will soon. DNA makes RNA, and RNA codes for proteins. So DNA to RNA is transcription because it's the same basic structure, AUCG, ATCG. It's the same language. So that's transcription. I mean, if I lost my job as a teacher, I might be able to do transcription. I'm not a great speller, but I'm pretty okay with English. English to English, okay. I could never be a translator, which is RNA to proteins. Translation. I'm lousy at foreign languages. I really am. Um, but the language of RNA is very different than the language of proteins. This one has 4 letters, AUCG. This one has 20. Very, very different. So transcription factors are molecules that can tell DNA to go, to turn on, or in this case, to turn on. Okay, so DNA makes RNA as transcription, and then just for the sake of completion, I can draw the ribosome. Here's the messenger RNA, and that would be making a protein. Okay. So examples of hydrophobic messengers or ligands are steroids, testosterone, estrogen, progesterone, aldosterone, or the thyroid